Good morning, everyone again. Welcome to today's next talk bioinformatics webinar, Human Whole Genome Sequencing. Thank you all for joining us today. I'm Minty and I am the moderator. To make sure everyone has a great experience, here are a few things to know. This session will run for about 40 to 50 minutes and we will have a Q&A session towards the end. If you have any questions during the webinar, please feel free to submit them through the chat box and we will address them after the presentation. Lastly, please keep your mic muted throughout the session. In today's next talk, we are happy to welcome Ms. Jiaxun as our speaker. Jiaxun is the bioinformatics manager at NextGene Scientific. She has over a decade of experience in her field. Today, she's going to walk us through how we can analyze human whole genome sequencing data using the bioinformatics tool EPI2ME. Without further ado, let's start the webinar today. Jiaxun, the floor is yours now.
Hello, is my mic okay?
Yes, your mic is okay.
Okay, so let me pull out the slide here.
And we can see your slides.
All right, thank you. So today, let me adjust my screen a little bit. So I'm using two screens over here, so you might see me switching here and there. Okay. So throughout the session, if you have any questions, please feel free to drop the questions into the chat box. And then because this human whole genome sequencing and the data analysis is relatively big topics, so I won't be able to go into the very details of the whole pipeline and the analysis. So if you have any questions after this, you can also reach out to us to go through the more details of the pipelines. So without further ado, let me start for the session. So today, before we go into the human whole genome sequencing data analysis, we will be looking kind of rehash a little bit on the nanopore sequencing, particularly the Oxford Nanopore Technologies and also their devices. And then we will jump straight into the EPI2ME for the data analysis. So introductions to nanopore sequencing. So this nanopore sequencing basically is the inventions from Oxford Nanopore Technologies. So Oxford Nanopore Technologies, or in short ONT, is a UK-based company. And then they have multiple solutions for DNA sequencing. So this DNA sequencing technology is rapidly gaining traction since the application is developed. So you can see it's reflected in more than 14,000 of scientific publications and adoptions in more than 135 countries by the end of the year of 2024. By now, it should be more than that. So in the slide here, you can also see the devices available from ONT for DNA sequencing. So the sequencing have been applied or used in many different communities, and it has been tested in many environments, including the space as well. And then it has been used in many also different industries by different users for different purposes. So it's a very versatile and very flexible technologies. So as long as you have samples where you can get the DNA sequence or the DNA samples out, most likely you won't have any problems to do the DNA sequencing by using the Oxford Nanopore Technologies. So why Oxford Nanopore Technology? So the ONT platforms is actually very different from other sequencing technologies. And then it actually can help to get richer insights. So this is also the only sequencing technologies that can sequence directly the native DNA and RNA molecules. So you basically, because in our DNA samples, the DNA molecules strain normally also could have some epigenetic modifications. Some of the bases, like the cytosine, could have the methyl group attached to it, which control the expression of the genes by either protecting the whole molecules or kind of after the methyl script is kind of removed, and then the native DNA molecules is exposed to the transcription factors, then the expression of the genes can be happens accordingly. So because Oxford Nanopore Technologies is able to sequence the native DNA and also the RNA, it can capture the epigenetic signals from the DNA molecules. So with that, we can also analyze, in addition to the genomics, we can analyze the epigenetics of it. And then it also provides faster time to results. So that means the real-time DNA sequencing means we are able to get the data out and also running the analysis while the sequencing is still ongoing. So unlike other sequencing technologies, we normally put our samples to the sequencing device, and then we need to wait until the sequencing process is done. Then we can get the data out for analysis. So this is different in ONT. So ONT, while the sequencing is still running in the device, we already can get the data out, and then we can see whether the data is something that within our expectation. If let's say we are seeing there are sufficient data obtained from the sequencing run, we can stop the sequencing anytime by looking at the real-time sequencing process. Also, all the devices from ONT are relatively small compared to other sequencer. So it's kind of easier for users to place it in different labs. And also, if let's say any relocation is required, then it's also very easy to relocate to different places. So because the sequencing device is so small, I think the smallest is just smaller than my palm. So you can bring it over to the field for sequencing as well, if let's say that's your preferred ways to do the sequencing. So how the nanopore sequencing work? So this is the core of the technology. So from the name, the nanopore. So the nanopore basically is a proteins that is in the shape of like with holes within the proteins, where it's very small, but it's still sufficient for a single DNA or RNA strand to go through the pores. So because DNA and RNA strands or molecules, they are carrying charges, the electrical signals. So when they pass through the nanopore sequencing device will be able to capture the signals changes when the process is happening. So that is why we are able to kind of real-time identify the bases within the DNA molecules or RNA molecules while the sequencing is still ongoing. And then because this process is controlled by the ONT device real-time, so in that, that also means that we are able to control if, let's say, the nanopore is capturing some DNA molecules that is off target. So that is something that we are not within our target regions. So the process is called adaptive sampling. So we can tell the software what are the sequence or what are the molecules that we want to look for in our DNA samples. And then the software will be detecting the DNA molecules while it passes through the pores. And if let's say the DNA molecule is not something that within our target, then the software can send the signals to the nanopore and then ask the nanopores to eject the DNA molecules out from the process and then capture another DNA molecules for the sequencing. So let me show you the animation of it. So this is how the DNA sequencing happens inside the Oxford Nanopore sequencing flow cell. So the pores are fixed to a membrane where the DNA that we prepare, when we prepare it into the libraries, we are providing it, kind of attach it with a motor enzyme. The motor enzyme will bring the DNA or RNA molecule all the way to the pores, the nanopores protein. And while this happens, So while this happens, then the motor enzyme will unwind the DNA, the double-stranded DNA molecule, make it into single strand, and then let the strand to pass through the pores. So while the DNA molecule pass through the pores, the electrical signals will be gathered, and then this signal will be turned into base calls by using a process called the base calling. So the whole process is like we sequence directly the DNA molecule, and then because the DNA molecule or RNA molecules are sequenced directly, so we do not really include the PCR process during the sequencing experiment. So that also means that the whole data that we are gathering are kind of PCR bias free. And then even though nanopore sequencing is identified as long read sequencing, but actually it can also sequence very short sequence all the way to very, very long sequences. So real-time analysis, while the sequencing is happening, we can already gather the electrical signals and turn them into base calls, and then we can obtain the data while the sequencing is still ongoing. So this is adaptive sampling. So where we are providing the information like what are our target regions in the DNA samples, and the software will control the whole process. So it will try to base call and then align the base call sequences to our targets and see whether it hits to our region of interest or our targets. If let's say it hits to it, then it will continue to do the sequencing. If not, it will eject out the DNA molecule and then try to sequence another one and test again. So this process is called the adaptive sampling. This is also quite oftenly been used for human genome sequencing, especially because human genome is relatively big in terms of their genome size. It's 3 GB or more than 3 GB. So sometimes we do not really want to sequence the whole 3 GB of it. We just could be targeting maybe a few hundred of genes. We want to have a quick look into those genes while we are sequencing the whole genome. Then we can inform the software what are our target regions. Then it will try to enrich the target regions instead of we design primers for more than 200 or 300 genes or probes to capture the target regions. So the sequencing device, basically we have two different flow cells depending on the throughputs or data throughputs that is required. For human whole genome sequencing, normally we will be using the ion flow cell, which is on the right side on the slide. So this one can provide 100 GB of data per 100 GB to 200 GB of data per sequencing run. So compared to the main ion is only providing around 10 to 20 GB of data per sequencing run. So because human genome, especially the human whole genome sequencing, normally we are requiring around 30x coverage. That means 3 GB, we are going to sequence the 3 GB at least 30 times. In total, normally we are expecting around 90 GB of data. So that's why one ion flow cells is estimated to provide us one whole genome sequencing data. So with the flow cell selected, the ion flow cell can be run or can be used for sequencing by using different sequencing devices. So at the moment we have the ion 2 solo or integrated. The solo, the P2S, this one do not come with the GPU, and then normally we also need to kind of get a computer that is of high GPU power to help with the sequencing process. And then this device will be discontinued soon. And then now, if sequencing, normally we will be looking into the ion 2 integrated, which come with a GPU and also the compute power. So with the P2I or the ion 2 integrated, the sequencing the device can use to cater two sequencing run at the same time. And then if let's say more samples are foreseen in a project, then we can also look into this ion 24. It can handle 24 flow cells at one run. So after the sequencing, then it will come to the analysis part. So from ONT, in addition to the sequencing solutions or technologies, they also provide the software or the program that is used to handle the sequencing and also the data analysis. So for bioinformatics analysis, normally we have two categories of programs. So one is with graphical user interface, so which is point and clicks. And then another type will be command line. So a lot of people think bioinformatics is quite challenging because it requires the skills in command lines. But for nanopore sequencing, normally the users can use some software that comes with graphical user interface. That also means that users do not really need the command line knowledge in order to do the analysis. So the software that comes from ONT, there are two. One is MinNode, another one is EPI2ME. So MinNode is the software that helps us to control the sequencing or to monitor the sequencing status. And also the one that we normally use for doing the base calling. And then the EPI2ME software is the one that we use for the data analysis. So this basically the software MinNode and EPI2ME, in addition to the base calling, the QC, the analysis, they also provide the reports after the analysis is done. So of course, for users who still prefer to use the command line tools, the EPI2ME workflows or the pipelines are also available in the command lines. And also we have the ONT or nanopore sequencing. There are a lot of users right now. So the tools or the software that are available for handling the nanopore sequencing data, there are also quite a lot of them. So most of them will be on kind of open source program and have freely to be used by other users. So for this session, I will be looking slightly more into this base calling process, because for human whole genome sequencing, one of the kind of the benefits of using the Oxford Nanopore sequencing device to do the sequencing is that we can capture the genomic data. We can also capture the epigenomic data at the same time. But this epigenomic data, it actually requiring some settings on the base calling part, either the MinNode or the Dorado. So this is the interface of the MinNode. So normally during the sequencing, we will be enabling the live base calling to be carried out. And then for human whole genome sequencing, if the epigenomics is something that you are also looking at, this is the options that we should be enabled during the sequencing process. Then the output will be in the BAM file, where it will also capture the epigenetics signals from the sequencing run. If we forgot to turn on these particular options during the sequencing run, we are still able to reproduce the modified base information from our data once the port5 file are available. So not to worry that if, let's say, this is not really turned on, as long as you keep the port5 or the raw signal files from the sequencing run, we are able to redo this process and then to measure the epigenomics once the data is available. So back to the analysis part, the EPI2ME program. This program basically can be installed in different operating systems. So if you are having Linux or having Windows or even the Mac OS, there should be no problem to install the EPI2ME program in your computer. So like mentioned, it comes with graphical user interface, is point and click. So we do not really need any command line knowledge in order to run the analysis by using the EPI2ME. It comes with pre-configured workflows or pipelines. So that also means that we do not really need to identify what are the steps that are required to do the analysis. So some common pipelines have been established. So we just need to follow through or select the right one in order to process our data. So ONT also provide a free cloud resource in case the computer that we are having is not high power enough. But for human whole genome sequencing, because the data involved is human related, and then normally the human data will kind of involve some privacy and confidential issue. So the cloud resource provided by ONT is not available for analyzing the human data. So for human data, we still need our own computer to do the analysis by using the EPI2ME. So the workflows or the pipelines that are available in the EPI2ME, in addition to the human genetics related pipelines, at the moment there are three: trio analysis, human variation, and also somatic variations for cancer study. And then in addition to the human genetics, there are also pipelines that are available for analyzing different applications, such as the transcriptome, the targeted sequencing, the metagenomics, bacterial genomes, fungal genome, and etc. So depending the needs. So today we will be looking deeper into the human variation pipeline. For this pipeline, it will get the kind of use the output from the sequencing run, basically the output from the min node as its input. And then after running through the human variation pipelines, what we are going to get will be the variants, human variations. That includes the small nucleotide variations, structural variations, short tandem repeats, expansion, copy number variation, the methylation or the epigenetics information, as well as during the phasing. So in some cases, we would like to also get the kind of want to know whether the variations come from either side of our parents. In normal experiments or short-read sequencing, we normally need to kind of do 3 samples of sequencing, the trio samples, the parents, and also the proband, in order to do the haplotyping or the phasing. Because Nanopore can handle long-read sequencing, so in that case, we are able to also perform the physical phasing. That means based on the reads. So as long as the variants come from the same single DNA molecule, we are assuming we will be putting them into the same haplotypes. So in that case, we are able to separate out different alleles by their haplotypes. So this is also one way, kind of one of the very good benefits from using the long-read sequencing. So after this pipeline, reports will be included at the end of the pipelines. So, and then the analysis results. If you are familiar with other human whole genome sequencing technologies like short-read sequencing, then the output from this pipeline will be very similar to those as well. So you should have no problem to use your preferred downstream analysis tools like the IGV or maybe cube variants and etc. to handle the data or results coming out from the pipelines. So this is the whole workflows, how it's going to look like. So during the sequencing and base calling, we normally will be using the MinNo or the engine is called the Dorado to do the base calling and then enable the methylations if, let's say, we are also looking into the epigenomics. And then the output, supposedly, the port 5 file will be the very raw signal files. So these files are relatively big in file size. So it will take some storage space to keep the files. And then once the port 5 file is available, we can do the base calling if, let's say, we prefer to use a more updated models and etc. to obtain our sequencing or the sequence data. And then FastQ can be also obtained. But if, let's say, we are looking into the epigenomics and etc., normally we will recommend to output the BAM files. So this BAM file basically can capture the sequence together with the sequence quality and together with the methylation information for secondary analysis. So once the sequencing and the base calling process is done, with the BAM file as our input, we can use the EPI2ME human variation pipeline to do the analysis. So this analysis include the steps that we have mentioned just now. So to be more details on what are the analysis that will be carried out. So this pipeline is expecting the BAM files as the input, and then we also need to select the reference genome to be used in the pipelines. So though depends on the projects, most of the time we will be using the GRCH38 or the HG38 as the reference genomes, but some of the legacy projects or projects that we need to compare to our maybe previous data sets, it could be coming from the HG19 or the GRCH37. We are also able to select the reference genome accordingly. So the steps involved, it will map the sequencing data against the reference genomes that we select. From the mapping or the alignments, it will kind of come up with the stats, how is the overall alignments looks like. And then from there, it will proceed to do the SNP calling. If let's say we are looking into the small variants like the single nucleotide variation and also the small indels, small insertion deletions, it will be running the analysis if this is our one of our objectives. In addition to that, it will also proceed to do the modifications analysis if let's say that is enabled. And then other pipelines or sub-workflows included will be the short tandem repeats expansion detection, the structural variation, and also copy number variation. So each of them, it will come up with its own sets of results together with the reports. So let me try to show you how the things looks like if let's say we are going to run it in the EPI2ME. So in the demonstration later, I'll be showing you the compute requirements, the input, and the reference files. How are we going to prepare the input files and etc. And then the whole pipeline of it, how we run the analysis, and then how's the output looks like, and then how we can further visualize the results. So for these human variation pipelines, the recommended compute power will be 32 CPUs and 128 GB of RAM is actually quite high. Is kind of higher than the normal personal computer that we would have for our routine works. So if let's say that is too high, I think minimum as long as the computer has around 16 CPUs or threads, and then with around 32 GB of memory, the pipeline still would be able to run, even though it might take slightly longer time in that case. So one thing to note that is ARM processor support is forced over here. So that means ARM processor, likely they are not able to run the analysis. That would include the computer, maybe like the Mac computer with maybe M3 or M4 processors, then likely this pipeline will not be able to, it's not so compatible with the Mac computer at the moment. So yeah, we will need some computer that with Intel or AMD chips with around 16 CPU and 32 GB of RAM. So for the demonstration, I'll be using a very small subset of data. So this is how the input files that's supposedly to looks like. So we can provide the pipelines with one single BAM file for one samples, or alternatively, because ONT is running the sequencing and then doing the live base calling. So that means every 1 hour or less than that, we are perhaps obtaining one BAM file from the sequencing run. So kind of once the sequencing is done, we might have multiple BAM files from one sequencing run. So it is also okay if let's say we provide the pipelines with a folder that is containing multiple BAM files. So these two basically are okay to be used as the input as for the pipelines. So in terms of the reference genome, the recommended reference genomes to be used will be the GRCH38 and also GRCH37. So the link is also provided in the EPI2ME, the pipelines documentation. So we will be looking into the link later. So yeah, And then the demo data set we are going to use for today, actually it's a very small data set. It only contains a small regions from chromosome 6, chromosome 20, and also the mitochondrial chromosomes. So it's actually a relatively old set of data. So compared to the recent data that we've seen, I think starting maybe this year, I think we are seeing the quality of data and throughput of data to be much better compared to few years back. So the analysis that we are going to try out, we will be doing the kind of calling the SNPs, calling the structural variations, and also calling the modified bases. And then we are also enabling the phasing. So that means we are going also to look into the different haplotypes of the same data sets. So yeah, we will come to the example reports for doing the analysis. So this is how's the EPI2ME program looks like. So in order to get the EPI2ME program, let me quickly bring you to where we can get it. So the site that we can download or obtain the EPI2ME program is over here. And this program is actually free for everyone to use as long as you have the nanopore sequencing data. Feel free to get a copy of this particular program installed in your computer. Previously, I think they do not really share it publicly. I think now is actually already open for everyone to use. So when you are in this page, you will see some latest updates regarding the EPI2ME. So over here, you can see the download button. Just click on it, and then you will be to this come to this page where you can get the EPI2ME installer. So depending on the computer that you are using, so if you are using Windows, this is the installer that you should be getting. If you are using maybe Linux, different types of Linux, you can also get the installer accordingly. So Apple computer, normally there should be no problem to install the EPI2ME as well. Just that some of the pipelines in EPI2ME might not be compatible with the Mac computer, like the human variation pipeline. So installation, at the bottom here, you can see there's an installation guide. So how to set up the program, basically, supposedly is a very straightforward process, but to make sure everyone will be able to install it accordingly, the guide is kind of very detailed in showing all the screenshots, like what could a user encountering during the process over here. Yeah, so you just need to follow through the process, and then there should be no problem to install the program in Windows or Mac OS or in Linux. So once it's done, and then when we turn on the program, we should be it should look like this. So but actually before come to this page, there should be one login page, and then we have options to log in using a free we can register a free account at ONT site and then sign in EPI2ME. Alternatively, we can also continue using the program as a guest. So depending on your preference. So here I have logged into my user account. So normally after login, we will come to this dashboard. This is also the homepage of it, where we can see the latest updates or latest status of the program, like what are the analysis that have been run recently, and then how's the computer status at the moment. And then if there are any news from ONT, they will also put it over here. So, and then if let's say there's any more updated EPI2ME program available, or kind of notes will also be shown over here, and then it's up to user whether we want to update the EPI2ME to the latest version or not. And then from here, at the bottom left here, you can see a setting buttons. So these settings, normally we will be, it will help to control how the analysis is carried out in EPI2ME, including like where the outputs or all the analysis will be carried out. So this is the working directory. By default, it's going to be in our user account in the C drive, let's say we are using the Windows. And then normally I will also take a look at the local tab over here. So this for first thing I just want to make sure the system is ready for running any EPI2ME analysis. So it actually have two dependencies. So one is WSL, another one is Nextflow. So when they are not ready, we can do the setup over here. But these two programs, when the program is left kind of inactive for some times, they might go into the inactive mode or the sleep mode. So if let's say they are not showing the status to be green ticks over here, we can also click the open setup and then retry to wake it up or to refresh it a bit. If let's say the programs have not been installed during the setup, we can also proceed to do the setup over here. So the EPI2ME will help us identify the right version of WSL and also the Nextflow, and then do the installation accordingly if let's say they are not available yet in our computer. And then over here, we can also kind of control how much computer resources that we want to allocate for EPI2ME to use to do the analysis. So this one, depending on our computer power. So normally I won't suggest to use everything, to use up everything for EPI2ME, because normally the Windows also requiring some RAMs and also some CPU to perform some routine tasks. So normally I will leave it maybe at least 4 GB to 8 GB for the normal window processors. So yeah, that's how we kind of look into the settings. So there are also other information available at the settings page over here, but it's okay. I think for most of the analysis, they are not so crucial for users to do the analysis. So we are going to skip other information over here. So other than the settings and then the home where we get back to the dashboard, from at the left panels over here, actually we only have two buttons left. So very straightforward. Launch button is when we want to do analysis, and then the analysis button is when we want to view the analysis results. So when we click into the launch button over here, all the pre-configured pipelines that comes with the EPI2ME will be listed over here. So here normally we just need to select the one that we want to use for our project. So there are pipelines that can be used for bacterial and fungal genome assembly. And then for today, we are going to use the human variation pipelines. In addition to that, there are also other pipelines available. And then you can see some of them is having the tag of community. That means the pipeline is actually not coming together with the EPI2ME. Is developed by other users, and then they share it. When they share their pipelines in GitHub, we also can import their pipelines in EPI2ME to do the analysis without requiring to run through the command lines. So and then for each of these box, one box represents one pipelines. So each pipelines, you can see two icons at the bottom right corners. So one is the computer icon, another one is the cloud icon. So when the computer icon is light up, that means the pipelines is available in our computer and ready to be used. If the cloud icon is light up, that means the analysis can also be carried out by using the cloud compute provided by ONT. And then there's no charge for using the cloud resource over here. So just want to show you, if let's say the pipelines is not available, if let's say it's grayed out like this, and then in case we want to use it, we just need to click on it and then proceed to do the installation accordingly. So back to the analysis pipelines page over here. So for human variations, the pipelines I have actually installed in the computer. So you can see it's already light up for the computer icon. The cloud one is not available because the cloud do not really accept human data to be submitted to their cloud. So that's why it's grayed out over here. So once click into this, once the pipeline is installed, we can have the full documentation of the pipelines directly in EPI2ME over here. So that include the kind of the brief introductions of the whole pipelines or what are the analysis that are included. And then normally I will also take a look at the pipelines overview, where it actually have all the step-by-steps, including the software that they are going to use, and then the version of the software and the parameters of the software, etc. So this is a very detailed documentations. And if let's say you want to just yeah, I think I need to show you here. Yes. If let's say you are going to get a reference genome to be used for the analysis, these are the two reference genome particularly for these pipelines. So actually I think both of them is stored in the Amazon cloud by ONT. But if let's say you prefer to download it from the public database like the NCBI, is also okay to get the reference genome from public database, just to make sure the pipelines actually the recommended reference genome is the one without the alternate context in the reference genome. So when select the data from NCBI or UCSC or Ensemble, it's better to choose the one without the alternate context, because that the alternate context could is going to make it complex when we want to map our data against reference genomes that have kind of multiple alternate contexts, where the software might have problem to identify where the rates should be mapped to when there are multiple sequences that are very similar in terms of their sources. So this is where we can get the reference genome. And then if you want to just rehash on how we should prepare the input, is over here. And then over here, the output, it also show after running the pipelines, because we are going to run multiple analysis, and then each analysis is going to give multiple output files. So here there's a kind of very detailed description on what are the files that we are going to get, and then the description for each of them. So in case we have any problems or the questions, we can also check out their troubleshooting guide and also the FAQs over here. So this is the whole documentation of the pipelines. And then on the right side over here, before we click the launch to proceed with the demo, I just want to show you over here, there are also options to manage the workflows over here, including running the demo analysis, check for the workflow or the pipelines updates, and then switch different versions of the pipelines. So the workflows that I'm going to use today will be the version 2.8.0. And then once we are kind of want to run the analysis, we just need to click the launch over here. Then we will come to the page of the setup page. So this setup page, on the left side, you can see all the settings or all the parameters that are available for the pipeline. Here, most of them, the parameters have been pre-selected or pre-configured and kind of optimized for the normal human WGS data. And then for us, we need to inform the pipelines what are the variants that we want to obtain from the analysis. So here, I think in this case, for the demo data, we are going to call out the structural variants. We also want to get the small variants, including the SNPs and also the small insertion deletion. Because our data is not the full genome, it's only part of the regions. So I'm going to skip the copy number variations over here. And then the demo data also do not really cover the short tandem repeats region. So that's why I'm also going to skip the short tandem repeats expansion detections over here. And then the data is coming from the native DNA sequencing. So I'm going to enable the modified base call to identify any bases, whether they are whether they are the modified base like attached with the methyl groups. So by default, it can identify the methylated C or the 5-HMC, the hydroxy methylated C. And then if let's say, I think human genome, less likely we will have the 6MA, the methylated adenine. But I think in some rare cases, I think that EPI2ME is also detected in human genome. So it depends on what during the base calling, what are the informations that we obtain from the base calling. So here it's going to detect whatever that is enabled over here. So other than selecting what are the variants that we want to detect, over here the main options, we need to tell the pipelines where are our input files, and then what is the reference genomes that we want to use. So here we just need to select our input file. So depending whether we have one single BAM file or we have one folder of BAM file. So here the demo data that I'm having is only one BAM file. So I'm going to select the BAM file over here. So I think I have the demo data in EPI2ME labs, the demo over here, human variation. So this is my input file. So it's a BAM file. Just select it as my input. And then in terms of the reference genome, actually they provide a reference genomes over here, but I just want to show you how the full reference genomes is going to look like. So I have one copy of GRCH full genome over here. So it should be a FASTA file, and then normally when we download, it's going to be a zip or a compressed format of the genome. So normally we will need to uncompress or unzip the file before we use it in EPI2ME. So I'm going to use this full genome over here. And then the minimum coverage. So normally the sequencing, we will try to get 30x coverage for the whole genome. But because this is only small data set, so I'm going to let this the program or the pipelines to do the base calling as long as there's one single rate covering the regions. So for data that either the targeted sequencing like the adaptive sampling or whole exome sequencing, or perhaps sometimes we might not have kind of achieved 30x coverage for the samples, then we can also adjust the coverage cutoff over here. So if let's say we do not really set the cutoff, we just want to make it 0 or make it 1 over here, then the pipeline will proceed even though some regions or our data set might not achieve 20x coverage for the whole genome of it. So I'm going to set it over here 1, and then the target regions. So let's say we are running some adaptive sampling or targeted sequencing or whole exome sequencing and etc. We can supply our target regions through a batch files over here. But because I'm just want to show kind of run whatever data that I have over here, so I will map the data against the whole genome of it and do not really select any target regions. It's okay to do so. So I'm going to skip the target regions. And for this data set, I also would like the pipelines to help me do the haplotyping or to phase out different haplotypes, different alleles. Then I enable the phasing over here. So other than that, actually you can see the settings or the setup over here all has been in green ticks. That means the pipelines is ready to be run. Before we start the analysis, just want to show you what are other options that we can adjust during the pipelines. So for copy number variations, by default, it's going to use another program to do the copy number variant calling. In this case, we are not running any copy number variant calling, so we can skip this. But in case you are having a low throughput sequencing data, or perhaps you are having adaptive sampling where you get kind of shallow sequencing for the whole genome, you can also enable the qDNA-seq to be used for copy number variation. And then for modified base calling, we can also ask the software to be kind of strand aware, so it will do the modified base calling or modified base identification, kind of separating out the forward strand and the reverse strand. And then we can also this short tandem repeat expansion, we are also not running these particular sub workflows over here. But in case you are running, you can additionally tell the software whether your sample is a male or a female. If not, it will try to infer from the data by looking at the coverage of the chromosome X and Y. And then other than that, we can also ask the program to enable the GVCF. So if let's say we have multiple samples and then by the end of the analysis, we want to perform joint variant calling across different samples, then we can also enable the global VCF file or the GVCF file to be part of the output. And then other than that, whether we want to downsample, if let's say we have really a lot of data obtained from the samples, then we can do some downsampling. And then the output format for the alignments by default is going to be the CRAM format, but I think some of the users still prefer the BAM format. So depends on your preference. You can also adjust accordingly. And then there are also some other parameters available for adjusting for different purposes. So feel free to optimize according to your project's requirements. Other than that, by default, it's going to use around 4 to 8 CPUs or threads to do the analysis. And then for different steps, they are allocating different numbers of CPUs or threads to do the analysis. If you would like the process to be faster, you can also increase the number of threads or cores over here for the pipelines so that more computer resources is allocated to do the analysis. And yeah, so also that one would depends on the the availability of the computer power. Other than that, I think I'm going to skip this. Yeah. Last part will be the run name. So by default, EPI2ME will assign a name to every runs in the EPI2ME. By default, it's going to be some random words. So if we want to make it more readable for ourselves, we can also kind of get a name for the analysis run. So for example, like I'm going to name this as demo on 18 June. Yeah, so that's it. So basically after selecting the variants that we want to obtain from the pipelines, and then what is our input files, and then I'll give a name, then supposedly there should be no problem to do the analysis. So to start the analysis, we just need to click the launch workflows at the bottom right corner over here. And then click again to confirm the analysis run. So once we click on it, then the software will help us to check whatever, all the kind of whether the computer is ready, and then what are the information that it needs. So it will start to prepare the analysis to be carried out. So the whole process is controlled by the next flow, which is a kind of a job scheduler. So as long as we allocate sufficient computer resources to EPI2ME, then actually we can run multiple runs analysis over here. The next flow will help us control the resources, the computer resources used for the analysis. So it's okay to, let's say you have multiple samples, it's okay to just submit everything into EPI2ME, and then the next flow will help us to control how to allocate the computer resources for each of the analysis. So it won't crash the whole computer or crash the analysis, but it will take some time for the, if let's say we submit multiple samples, then next flow will try to allocate the computer resources accordingly. And then the analysis overall, it might take longer time because the computer resources and ultimately is shared across multiple samples or multiple runs in that case. So, but we don't need to worry about how to whether the computer is going to be overloaded by certain samples or by certain runs or not. So everything controlled by the next flow. So from here, after the analysis has started, you can see the task or the process that is running at the moment. So now it has passed, kind of finish up the several tasks, and then now it's at the task of preparing the reference. So we will kind of, if you want to check the status of the analysis run, normally we can come to this page. So other than that, we can also look at the status, overall status at the top over here. So to check the analysis that has been carried out or is being carried out, we can click on the analysis button over here. Then it will show whatever analysis that is running or completed. So we can see the status of the analysis over here. So the one that we have just launched is being carried out, is running at the moment. So for the same analysis, I have also done the one run before this. So I think last week I had also tried out once. So I think this analysis could take around 1 hour. So we are not going to wait until everything finished to look at the results. So I'm going to show you how the results looks like once it has finished. So you can see a lot of steps actually will be carried out during the analysis. So as long as we see all the green bars over here, that means all the tasks has been completed successfully without any problems. Once it's done, we can also see the reports over here under the reports tab. So over here, we have multiple reports. If you click on this, we can see the demo report for different sub workflows. So we have one report for the alignment, one report for the SNP calling, and also one report for the structural variant calling over here. So depending on which report we want to look at, we can switch it over here. So the first report is supposedly to be the alignment QC report, where we can see how much data that we have submitted for the analysis. So this is a very small data set. We only have around 3,000 or less than 4,000 total reads. And then because this is only aiming certain regions in the human genome, so the targeted regions is actually having more than 100x coverage, even though we have a very little number of reads. So over here, we can also see out of the 3,700 reads, how many of them is mappable to the reference genome, how many of them is unmapped to the reference genome. So out of this number, we have 15 reads that cannot be mapped to the reference genome. So the rest have been mapped to it. And overall, the quality is like around, I think the peak is at around 19 or 20. The latest chemistry, I think we are achieving almost 25. And the read length for this run basically is 15 KB to 16 KB. And the data that we are having is roughly, I think, 60 MB. So it's a very small data set. And then after doing the mapping, how's the accuracy looks like, and then whether the reads are mapped fully or they are mapped only, half of the reads are mappable to the reference genome, and then half of them is considered unaligned. So the details of the alignments are all shown over here. So this data set is going to map to chromosome 6, chromosome 20, and also the chromosome, the mitochondrial chromosomes, but we are kind of left out the mitochondria over here. So chromosome 6, you can see the coverage for very small, tiny regions, and then the chromosome 20, the coverage is also only on small regions of chromosome 20. So, and let me switch it to the SNP report. So for this data set, it has called out more than 1,000 small variants, including like 900 plus SNPs, and also 100 plus of insertion deletion. So the overall transition transversions rate or ratio is around 2.3. So this is the more details on what are the SNPs that have been called. If let's say there are any SNPs or indels that have been called out and then they identified to be pathogenic or having some impact to the samples, there's a ClinVar variant annotation over here. If let's say that there's any interesting one, it will also be highlighted in the report. But for this data set, we do not really have any kind of pathogenic or kind of annotated variants on the targeted regions. So that's why it's no ClinVar sites to report over here. And there's the substitution tabs, summary, the insertion, deletion, length summary, and etc. So these are kind of a quick summary on what have been obtained for this sub workflow. Other than that, I think we also have a report for the structural variations. So how many insertion, how many deletions have been identified, and then how many chromosomes is having the structural variations. So here, I think both the chromosome 6 and 20 are having some structural variations. So over here, it also provides us with some summary of the structural variants that have been called out. And then this one, it will highlight where is the locations or where the structural variants have been identified. But because we are only having very, very small, very kind of structural variants over here, so it's almost not very visible over here. So if let's say we are doing whole genome sequencing and then detecting the structural variants across the whole genome, then we will be seeing more interesting plots over here. And then here will be the kind of the insertion deletion size distribution. And then for each end of each report, we have the very details like what are the software that has been used together with their versions, and also what are the parameters that have been used for certain analysis. So these few are actually only the summary reports. For the output files, we can see the output files over here. So for each of the analysis that have been carried out, it might have more than one output files. So we can cross-check with the documentations on what are those files by kind of the general one that is important for us. Normally will be the alignment files. And then for the reports actually is provided in the HTML format where we can share it with our collaborators or our lab mates to also look into the results. And then for the modified base analysis, the report is not available, but the output file is over here, so we can see the modified base big width file and also the metals back file. So the files are over here. And then we also have the SNCs file in the VCF, the structural variance files in VCF, and also some additional files. So here we can get the output file from this page, but normally I will just open up the whole folder of the output file by clicking the options on the upper right corner and then open the folder. Then it will open up the file explorer where all the output files are stored. So basically these are all the output files, and then we can further work on them, kind of further analyze on them accordingly. So these files normally for human whole genome sequencing, the data, the results is going to be very big and a lot. And then normally we will need some maybe further tools, genome browser to help us to look into them. So this alignment file, I think in CRAM, and then the VCF, the methylation files in the big width file, all these are acceptable as the input for downstream pipelines, or they are actually following the standard format. So if let's say we prefer to use certain downstream program like IGV, the genome browser, we can actually open up the files accordingly in the IGV. So this is the CRAM files where we can see the alignments over here. And then on the top parts, we can also see the coverage for the, I think now we are at chromosome 6. And then this particular DUSP22 genes. So we can see this gene has been covered fully, including the intronic regions. And then if let's say we are looking into certain SNPs, we can also put in the SNP VCF into the IGV. And then if we are looking at the modified base signals to see whether any signals or any regions are highly methylated or kind of relatively hypomethylated, then we can also put the signals files over here by loading the big width files. So here you can see some of the regions are methylated, some is not, and then some is only methylated at a certain strand, but another strand is not really having any methylations. So, and then if we have multiple samples, we can also put in multiple samples into the IGV to do some quick comparisons. And then this one, of course, we can also zoom in and out accordingly to look at the alignments, look at the variations, and etc. So a very light version is also provided here within the EPI2ME. But because this one is a very light version, so it might not have the full functions like what we see in the full version of IGV. And I think that is where we can get all the reports and output file. And then at the last tab over here, there's a log file as well. So this normally it could be kind of long and then not so meaningful for most of the users. But normally this is where we can do some troubleshooting in case the analysis run into problems. So let me try to get some analysis that start with error. So in case some of the analysis run into problems, and then this is the place that normally we'll try to see what happens, what are the cause of the errors. So this one, most of the time is still not so kind of user friendly. It might not help directly to kind of help us to identify the problems. So one way is we can copy the whole error message to AI like ChatGPT or Gemini or any AI tools that you prefer. You can copy the whole message of it and then put into the AI, and then tell the AI that you are running EPI2ME and then which workflows that you are using, and then the error is like this, and then ask the AI to help to troubleshoot and then suggest how we can rectify the errors. So this is one way of doing the troubleshooting. But in case the AI is still not able to help us in troubleshooting or give us a good solutions on that, you can also try to click on the report issue over here on the fail run. So once it click into it, it will show you the step on how to proceed with the error by submitting the case to the Nanopore tech support. So once we export the whole analysis log files, we can send the log files to support@nanoporetech.com, then through the email. And then in the email is also, we need to describe the problems that we are having, and then give as many details as possible or the context to the Nanopore tech support team to help with the troubleshooting. So once we click on the export, it will actually help to wrap up the log files and then the important files for us to kind of do the troubleshooting. So this one we can send to the Nanopore tech team to help on the troubleshooting. Alternatively, you can also reach out to NextGen or reach out to me in case you need some help in terms of troubleshooting the analysis that you are running. So yeah, this is how we handle the analysis in case it failed. But most of the time, if we follow through the process, telling the software what is our input file, and then the input file is from Nanopore sequencing according to their protocol, most of the time we won't have any problem. But in case there's any problem, you just there are ways to do the troubleshooting. Okay, the one that we run just now has completed. I think it takes much faster time than what I have been expecting. Yeah, so the reports is over here, like what we have seen just now, and then the output files, and then the viewers, and then the log files. Yeah, so it seems like everything is running okay. Yeah, because our data is actually not having sufficient coverage, so it will not really helps to further do the analysis because the coverage is simply too low. All right, so that is what we have for the demonstration. So if we get back to the slide over here, so these are some of the screenshot of the reports that we are going to get from the human variations analysis pipeline, including the alignment reports. And then the small variation reports and structural variation reports. And also, if let's say we are also running the copy number variations, we are going to have a reports together with the predicted karyotype if let's say we do not really inform the pipelines the gender of the samples. And also, if let's say we are also interested in looking into some short tandem repeats expansion, so in human genome, normally there are a lot of short tandem repeats, like certain units like GGC is going to repeat for 20 times. So normally repeating for 20 times or less than that, it should be normal. But certain repeats like this one, the AAAG in the chromosome 4, if let's say it repeats, the repeat number is a lot, like more than 100 copy of it is going to be a pathogenic repeat, so it causes some problems. So this tandem repeats expansion is also kind of the pipelines is available inside directly in the same pipeline. So in case you are interested, you can also enable the analysis to be carried out. Yeah, so the output file it looks like this. So depending on the file names, I think we can also already guess what are the data that is captured within. But to double confirm, you can also check out the documentations to have a more details on each of the output files. So with this, basically today we have gone through briefly on the ONT and the nanopore sequencing, and then how we can get the EPI2ME and then do some analysis using the EPI2ME, particularly on the human variation pipelines. And also we have gone through the results and reports together. Even like the downstream like IGV, we can also get the visualization to be done using the output from the EPI2ME as well. So this basically the end of my demonstration and presentation. So thank you so much for your time.
Thank you, Jiaxun, for the insightful presentation. Now we will move into the Q&A session. So you can unmute yourself or you can type in the chat box. Is there any question from the floor? Let's wait for a couple of seconds. Yeah, it seems we do not have any question from the audience. So let me conclude this session now. Thank you all again for participating in today's Next Talk. Next, we would like to hear your thoughts for today's sessions. If you have a quick moment, please fill out the survey in the chat and we will send the certificate to you. If you have any question after this Next Talk, feel free to contact us at sales@nextgen.ny. And thank you all once again, and we look forward to seeing you again in our next webinar. Bye-bye.
Thank you.